Dear viewers, today in this lecture we would be learning about the transport systems of plasma membrane. In the year 1665, Robert Hooke first reported the existence of cell. What Hooke really saw in the bark of the cork oak were empty, dead cell walls, devoid of the living matter they once contained. Other early microscopic observations also described cells in all kinds of plants. It turned out that animals contained similar unit, but these were harder to see because animal cells lack the cell walls that surround plant cells. Instead of cell wall, it has dynamic phospholipid bilayer as a outermost covering. Same kind of structural arrangement is also present in plant cells beneath the cell wall, known as plasma membrane or cell membrane or plasma lemma. The plasma membrane. To remain alive, the cell must maintain chemical homeostasis, so it must keep its internal chemical composition constant within the narrow limits suitable for life. However, the cell cannot simply create a suitable internal environment and then seal itself off from the world to avoid gaining or losing substances by diffusion. The biochemical reaction of metabolism requires raw material from outside the cell and generate waste product that must be expelled. Hence, the cell must maintain homeostasis while continuously exchanging substances with surrounding environment. Control of what substances enter and leave the cell is the task of plasma membrane. The plasma membrane is selectively permeable, that is, it permits some substances to pass more freely than other and even prevents the passage of certain kinds of molecules to which it is impermeable. Hence, the concentrations of the ions, nutrients and other substances inside the cell differ from those in the cell's environment. In recent years, our understanding of membrane and how they work has progressed rapidly. We now know that all biological membranes broadly have rather similar structure and function, whether they are plasma membrane or internal membrane of cellular organelles. On the other hand, the dry weight of all biological membrane consists of lipid, proteins and carbohydrates. The actual kinds and proportions of these molecules present depend upon the membrane, its location and function. Now we will study transport through plasma membrane. Many molecules, particularly polar molecules, move rapidly across biological membranes even though cross artificial lipid bilayers very slowly. Examples include various small ions, glucose and amino acids. These substances are transported by membrane transport proteins or carriers. Each transport protein is specific in that it transports only one or a few chemically similar substances. Some transport proteins merely move only one type of solute across the membrane known as uniport. In other, transfer of one solute depends upon the simultaneous transfer of a second kind of solute known as co-transport. If simultaneous transfer of two molecules is in only one direction, then it is known as symport, while if it works in opposite directions, then it is known as antiport. Depending on the requirement of energy to transfer the molecule through the plasma membrane, there are mainly two types of transport. Number 1, passive transport and number 2, active transport. On the other hand, depending on the size of transported molecules, membrane transport can be divided into micromolecule transport and macromolecule transport. The macromolecule transport requires energy and hence it comes under active transport, whereas micromolecule transport can be either passive or active type. Now, First, we will see the different types of micromolecule transport. Types of micromolecule transport. Depending upon the expenditure of energy required, the micromolecule transport can be classified into two types. Number 1, passive transport. It is movement of molecule from its higher concentration to lower concentration across the plasma membrane, either spontaneous or by mediating some carrier protein without expenditure of energy. Number 2, active transport, in which the transport of materials is across a membrane against an electrochemical gradient with the expenditure of energy. We will be studying each of these in detail, starting with passive transport. In this transport, the molecules of any substance move away from a region of their higher concentration to a region of their lower concentration. Such movement is random and it is according to the concentration gradient, hence energy is not required. This behavior is called diffusion. The greater the difference in the concentration between the two regions, the faster is the diffusion. Diffusion of gases is most rapid. Compared to it, the diffusion of liquids is slower. Such diffusion continues till the balance becomes established between the concentrations in the two regions. There are mainly two types of diffusion, A, simple diffusion, and B, facilitated diffusion. A, simple diffusion. Movement of hydrophobic molecules and some small uncharged polar molecules like urea, water, glycerol and carbon dioxide, that is CO2, across the plasma membrane depend upon their concentration gradient and is known as simple diffusion. The diffusion process can be understood easily by Fick's law. The law states that the amount of solute ds diffusing across the area A in a period of time dt is proportional to the concentration gradient dc upon dx at the point. The equation can be summarized as ds upon dt is equals to dA into dc upon dx, where d is the diffusion coefficient. There are two kinds of arrangement in plasma membrane for simple diffusion. Number 1, static pores. They form randomly through lipid bilayer as a result of thermal movement of acyl phospholipid chain. Hydrophobic molecules like benzene can pass easily through static pores. Number 2, protein channels or aqueous channels. Perhaps the simplest case of passive transport occurs where membrane proteins form channels through the lipid membrane. These either involve only one transmembrane protein or it involves clusters of integral proteins. These channels contain an aqueous solution and permits small solutes or small uncharged particles like water, that is H2O, urea, glycerol, etc. to cross the bilayer by simple diffusion down their electrochemical gradients by avoiding the membrane's hydrophobic interior. While some of the channels are open all the time, other called gated channels behave as if they have gates that open and close. There are number of gated channels like voltage gated channel, ligand gated channel or mechanically gated channel present in the plasma membrane. B. Facilitated diffusion. The movement of hydrophobic molecules or large uncharged polar molecules like glucose, sucrose, etc. or some ions cross the plasma membrane by mediating the carrier protein and is known as facilitated diffusion. It is a carrier assisted transport mechanism, and just like simple diffusion, it does not require cellular ATP or energy for its operation. The driving force here is a concentration gradient. but the movement of solute across the membrane is relatively more rapid than simple diffusion, mainly due to specific carrier molecules involved in this process. Carrier molecules which can transport the substances across the membrane is thought to be a protein. According to this line of thinking, a given membrane would contain a set of facilitator proteins, each one having a specific binding site. The carrier protein, after binding to its particular molecule, would have the task of moving the molecule to the other side of the membrane, either by a conformational change, rotation, diffusion, or by forming a pore. According to the Fick's law, the rate of movement of a substance is directly proportional to the concentration gradient, which holds for simple diffusion, but in the case of facilitated diffusion, saturation kinetics can be demonstrated. Here in the facilitated diffusion, plotting the graph of rate of entry versus concentration gives us asymptotic curve. An example is the system that involves the diffusion of glucose into the cells of some tissues of vertebrates. In the liver, the lens of the eye and red blood cells, the facilitated diffusion moves glucose across the plasma membrane in both directions by means of carrier molecule. The carrier molecule or glucose facilitator is more likely to encounter and pick up a glucose molecule on that side of the membrane where glucose is in high concentration. When a cell is quickly breaking down glucose during respiration, the glucose concentration inside the cell falls. Glucose is then more plentiful outside the cell and it is moved into the cell more rapidly than it is moved out. Cells in the liver, for instance, not only remove glucose from the bloodstream when the blood glucose level is high, but also replenish the blood glucose when its level drops. The glucose facilitator demonstrates nicely the specificity of the transport system known as stereospecific nature of facilitated diffusion. D-glucose has an apparent Km of about 10 times lower than D-galactose and over 1000 times lower than L-glucose. Km is the amount of substrate required to move the reaction at half maximum velocity. Thus, the lower the Km, there is higher the affinity of the molecules. Hence, the readily metabolized D form of glucose competes very effectively for transport into the erythrocyte even if present at lower concentrations than other hexoses. There are mainly three differences between simple diffusion and facilitated diffusion. Simple diffusion is relatively slower as compared to the facilitated diffusion. Moreover, simple diffusion does not involve any stereospecificity and does not show saturation kinetics, whereas facilitated diffusion involves stereospecificity as well as show saturation kinetics. Number 2. Active transport. Let us first compare the active and passive transport as well as see the difference between primary and secondary active transport. Two things distinguish active transport from the passive transport mechanisms. First, active transport can move substances against their electrochemical gradients, and second, active transport requires the expenditure of energy. The source of energy for active transport may be the high energy molecule ATP or the electrochemical gradient of an ion across the membrane. Common sources of such electrochemical energy are steep different in sodium ion, that is Na+, or hydrogen ion, that is H+ concentration on the two sides of a membrane. A. Primary active transport. Example of primary active transport is a calcium pump found in many cells which pumps calcium, that is Ca2+, out of the cells and so keeps their internal Ca2 plus concentration much lower than external levels. Another example of primary active transport is spectacular pump found in the stomach wall and is responsible for secreting stomach acids. It uses the energy of ATP and export H plus against a pH gradient of about 1,000,000 to 1. But one of the most widespread and best studied example is the sodium potassium pump. It uses energy from ATP to transfer sodium, that is Na plus, out of the cell and potassium, that is K plus, in. This pump is largely responsible for generating the electrical potential that exists across the plasma membranes of all animal cells. The pump is essentially a sodium and potassium dependent ATPase, an enzyme that hydrolyzes ATP in the presence of sodium and potassium. Transport of sodium and potassium ions is linked. In the plasma membrane, the pump moves 3 sodium out of the cell for every 2 potassium that moves in, and so the interior of the cell becomes progressively more negative compared with the outside. Furthermore, another membrane protein, the potassium leak channel, permits the passive leakage of potassium out of the cell down its chemical concentration gradient. This makes the interior of the cell even more negative. At some point, the tendency of potassium to leave the cell, moving down its chemical concentration gradient, is exactly balanced by its tendency to enter the cell because of the electrical gradient across the membrane. At this point, net movement of potassium ceases and cell is at its resting membrane potential, that is from -20 to -200 millivolt and negative inside depending on the species and cell type. Membrane potential exists in most, if not all, cells, but they play an important role in the working of nerves and muscles. The sodium potassium pump also controls the cell's water content and drives the active transport of sugars and amino acids. Remarkably, calculations indicate that 1/3 of all animals' energy expenditure goes to power this pump. The ability of muscles to contract, the digestive tract to absorb food, and the kidney to form urine all depend on the working of this ionic pump. B. Secondary active transport. Many times, transport systems are not powered by ATP, but by energy stored in the form of ion gradients across a membrane. For example, the active transport of some sugars and amino acids into animal cells is powered by the sodium gradient across the plasma membrane. All of these systems involve the dual movement of the gradient ion as well as of the substance being transported. As an example, the movement of glucose into cells of the intestine and kidney. Glucose and sodium simultaneously bind to different sites on their carrier protein, and binding of both these substances is necessary for transport to occur. The driving force for this transport is the electrochemical gradient of sodium. The greater the electrochemical gradient for sodium, the faster glucose moves into the cell. The concentration of glucose itself has no effect on the rate of glucose transport. Of course, the sodium that enters the cell with glucose in this way is pumped out again by the sodium potassium pump after expenditure of energy stored in ATP. In sense, the sodium potassium pump is responsible for this active transport of glucose because it maintains the electrochemical gradient for sodium that makes glucose transport possible. Hence, sodium potassium pump utilizes ATP as energy source and is known as primary active transport, whereas transport of glucose with the help of sodium electrochemical gradient is known as secondary active transport. Most ion-driven transport systems that occur in plants and animals are powered by sodium gradients, and other ions take the place of sodium in other systems. For instance, most bacterial active transport systems are driven by hydrogen gradients. As for example, the lactose permease system present in the plasma membrane of E. coli. The function of lactose permease system was first discovered by Ian West and Peter Michel in early 1970. They discovered that transport of lactose into bacterial cells is accompanied by an increase in the pH of the external medium. This observation suggested that the lactose permease might simultaneously transport or symport the protons and lactose molecules into the cell. Lactose permease is a highly hydrophobic integral membrane protein that contains a single lactose binding site and a single proton binding site. This protein spans the plasma membrane and behaves as if it exists in two conformational states, one in which these two binding sites face the cell exterior and one in which they face the inside of the cell. Transport is thought to be initiated by the binding of lactose and a proton to their respective sites on lactose permease. The binding of lactose and proton reorients the binding sites so that they face the cell interior, allowing the lactose and proton to be released within the cell. This dissociation triggers a conformational change that reorients the binding sites so that they again face the outside of the cell, allowing the cycle to be repeated. Now, we will be discussing the macromolecule transport. Studying macromolecule transport or membrane transport of large particles. Sometimes very large molecules or even bigger particles of matter must enter or leave a cell. Items of this size cannot pass through membranes either by penetrating the lipid bilayer or by protein transport mechanisms. However, the transfer of such large particles is possible because of the membrane's fluidity and dynamic nature, which permits it to change shape and to fuse with or pinch off small membrane enclosed sacs. If membrane enclosed sacs engulf material, it is known as endocytosis, and if the material is discharged, it is known as exocytosis. In the process of endocytosis, cells incorporate material from their surroundings. The unicellular organism like amoeba engulfs its food by extending projections called pseudopods from the cell body and surrounding the food. The pseudopods then fuse so that the food becomes enclosed in a membranous sac in the amoeba's interior. This kind of endocytosis is known as phagocytosis or cell eating. It is the engulfing of large particles approximately 1 to 2 micrometer in diameter, such as an entire bacterium or a fragment of a disintegrating cell, into a sac-like vacuole in the cell. Phagocytosis is a major feeding method of many unicellular and simple multicellular animals. In many higher animals, phagocytosis rids the body of debris such as dead cells and also plays a part in defense against disease. For example, some of the vertebrates' white blood cells are phagocytes which engulf and digest invading bacteria. Some of the body's cells such as red blood cells normally undergo programmed death and their remains are also cleaned up by phagocytes. Interestingly, the bacteria that cause leprosy and tuberculosis have adaptation which makes them resistant to digestion by phagocytes. In receptor mediated endocytosis, small items such as proteins or low density lipoprotein, that is LDL particles, are taken up selectively. For this to occur, the substance must first bind to specific protein receptors on the plasma membrane. The membrane then invaginates, forming a depression around a number of this receptor. The molecules that have bound are known as ligand. The membrane then pinches off a small sac containing the receptor ligand complexes into the cytoplasm. Low density lipoprotein particles taken into the cell in this fashion contain cholesterol and fatty acids for membranes. Pinocytosis or cell drinking is a kind of endocytosis in which a cell engulfs some of the surrounding extracellular fluid or liquid droplets of diameter approximately 0.1 to 0.2 micrometer. The plasma membrane invaginates, forming a narrow channel into the cytoplasm, and vacuoles pinch off at the channel's inner end. Pinocytosis is known to occur in amoeba and in some kinds of animal cells. Endocytosis does not occur in most plant cells because they are surrounded by thick cell walls which prevent large particles from coming into contact with the plasma membrane. In exocytosis, substances are discharged after the membrane of an internal vesicles or vacuole fuses with the plasma membrane, which then opens up. This allows the contents of the vesicles or vacuole to escape from the cell. Proteins can be secreted from cells by exocytosis in either a constitutive or a regulated fashion. In the regulated pathways, molecules are stored in secretory vesicle as in the case of hormones, or as synaptic vesicles in the case of neurotransmitters, which do not fuse with the plasma membrane to release their contents until an extracellular signal is received. Regulatory pathways of exocytosis operates only in specialized secretory cells. Constitutive secretory pathway operates in all cells, mediated by continual vesicular transport from the trans-Golgi network to the plasma membrane. There are mainly two functional significance of the exocytosis. First, it is used for the secretion of hormones, digestive enzymes and neurotransmitters. Second, it is also useful for throwing out undigested waste materials. At the end, we summarize the information which has already been discussed in this lecture. Cell must maintain a lively commerce with their environment, taking in new raw material for their metabolism and expelling waste products. On the other hand, it must maintain the internal concentration of all substances. The plasma membrane regulates the entry and exit of the substances in cell and is permeable to small molecules and ions, but sufficiently impermeable to prevent loss of nucleic acid, protein, lipid and polysaccharides. Membrane's lipid bilayer admits small lipid soluble molecules which diffuse through lipid layers according to their concentration gradients and is known as simple diffusion. Most ions and polar molecules cross the membrane with the aid of protein transport molecules. Other proteins mediate active transport that requires energy provided by ATP, known as primary active transport, or by an electrochemical gradient of ions known as secondary active transport. When the cell acquires macromolecules or larger particles, the membrane surrounds them and pinches off to become a vesicle or vacuole inside the cell by process of phagocytosis or pinocytosis, collectively known as endocytosis. On the other hand, substances can be discharged from many cells by opposite process known as exocytosis. So, either by passive or active transport mechanism, the plasma membrane continuously maintains cell homeostasis as well as functionality of the cell.